Alright guys, so we're kind of going to pick up right where we left off in class, and that's relating to the idea of what a secretor is and then what some of the presumptive blood tests are. And so the big thing you got to remember about a secretor is that you can determine the blood type from your bodily fluids. Saliva, tears, semen, any gastric juices. You create this extra, basically like this sugar antigen that can help determine what your blood type is. So the majority of us are secretors, which means you can determine the blood type, and then a very few small percentages isn't. And so that's what this is just all related to, is how we test it. And it's the same concept with blood typing. We use this protein-based antibody with your lectin. And if it clumps, then you know that you are a secretor. If it doesn't clump, then you're not a secretor. So that's just determining what your blood type is. Okay, then we got into different blood tests. And the big thing to know here about these blood tests is that they're presumptive, which means there's no guarantee that it actually is human blood. But we want to filter it out. We don't want to take 100 samples to the crime lab and have them all be tested for human DNA. We want to make sure that we only take the samples that tested positive for falling in this possible blood type group. So this one of the first ones we did, it's actually the most common one, and it's the Kastle-Meyer. And the reason why it's so common is because it's quick, it's inexpensive, and it is really, really sensitive to still being able to pick up blood. The biggest thing to understand about Kastle-Meyer is kind of the sequence of how it happens. So we're going to skip ahead here a little bit. And here's the sequence. And that's where you get, first you put the ethyl alcohol on there, and that's just to help clean it, to allow the hemoglobins to be more active and available to test. And then after you add the alcohol, then you're going to add the phenolphthalein. And phenolphthalein, the pH indicator, it is like oxygen starving. And as soon as oxygen reacts with it, it's going to turn a color, and it turns pink. Now, one way that we can speed this reaction up so we can tell right away if it's blood or not, and not just oxygen from the air, is by adding hydrogen peroxide. Hydrogen peroxide, it is a catalyst. It speeds up reactions. And when hydrogen peroxide meets up with the iron and hemoglobin on the red blood cell, it breaks it apart and produces oxygen and water. And so once that oxygen is available, then the phenolphthalein picks it up, and then it turns pink. So it's going to turn pink immediately. Now, one of the problems is if time goes by, we're talking 30 seconds to a minute, it will eventually turn pink because it's oxidizing with the oxygen that's in the air, and not because it's actually related to blood being on the sample. So this whole thing really just talks about exactly what I just went through. What the hydrogen peroxide does, what the phenolphthalein does, changing colors. And this explains the whole process as well. So, good thing to review and look through if you're still kind of on the fence of understanding what the Kastle-Meyer test is. Okay, next thing I want to talk about is hydrogen peroxide, and this is something you use in your house. It is a cleaner. And hydrogen peroxide, once again, it works with the iron, and it creates the white bubble foam. It's creating oxygen and water, and that's just a bunch of oxygen gas that's here. So if we move on, all of this is because of an enzyme that allows it to break apart the molecules found within the iron and hemoglobin. But you don't need to worry about the name of what that enzyme is. Okay? The biggest thing you need to understand is that it reacts with the iron on the hemoglobin, and it creates a white bubble foam. Okay? And it's an oxidation reaction, which means it creates oxygen, which is important when it comes to like the Kastle-Meyer test because it reacts and turns it pink. Okay, the last one we're going to get into, I'm actually going to skip ahead to look at some of these pictures, and it's luminol. Luminol is really cool. It does this chemolescence, fluorescent blue coloration, and once again, that's in the presence of blood or other unfortunate, we back this up, there's going to be other false positives. They have lots of false positives on this one, and that's bleaches used to clean up crime scenes. It could light up paints. I love that horseradish is on there. So there's lots of false positives when it comes to luminol. But if we look at this luminol, you can tell if this wasn't fluorescing blue, it just looked like normal, linoleum floor. But someone's cleaned it up. You can tell the bucket and what's going on around here. But even though it's been cleaned up, to the naked eye, it doesn't mean that the actual iron on the hemoglobins, those red blood cells, are completely all gone. And so it's still able to light up and react. The downfall, one of the downfalls of luminal besides false positives, is that it doesn't react for a long period of time. We're talking anywhere between seconds to 20, 30 seconds. And so they quickly take the pictures because then it fades. The reaction occurs and then it fades. Now, the good thing is you can reapply the luminol and it will react over and over and over, but it will keep continuing to fade, fade, fade. All right, so now let's go back. Like we said before, it's extremely sensitive. It's visible. You can see it. And when you look at this part, the intensity of the glow indicates the amount the blood was present. So I love that the luminol also shows quantity, like how much blood. Is it just a single drop, just a brush of blood, or is it a pool of blood? And like I said, there's definitely lots of false positives there. All right. So that's the biggest thing to know. And this kind of just goes through the process of how luminol works. So like we said, we know it's a presumptive blood test, and it'll be sprayed in a dark room, which it needs to, so it can fluoresce blue. And this occurs when it comes in contact, once again, with that blood. So it does, like we said, reacts to the iron found in the hemoglobin, and it'll last about 30 seconds, and then it's going to fade. So, and then, of course, there's always false positives. Anyway, so the thing that I like about this picture is you can see actually the torso, upper body, the head, the arms, and then it looks like this victim was drug out. Now, how bright, brilliant blue it is, it leads you to believe that they were badly wounded, because that's a lot of blood coming off and being drug out. And then looks as though the victim was sitting or laying on the couch, because you have an amount of blood that happened here as well. Now, the reason why I put these in here is because this is the exact same reaction that occurs within your fireflies and your jellyfish, is that cumulescence and reaction. Basically, it's like electrons moving and shifting, and it makes that brilliant, bright blue. This little link, and this would be an awesome one to watch. It just talks about how the process goes and kind of explains it in a little better detail. All right. Let's see what. Okay, so I moved on to the next slide after the 3 different presumptive blood tests, and you'll see this crime scene analysis of blood splatter. This is in your notes packet. This is on page 11. You can see it right here. Now, unfortunately, the website, the link has been discontinued, so we're not going to worry about this part. The crime scene analysis blood spatter. There's questions 1 through 5. You're not going to worry about answering them. Explanation of the images, and then it goes over to this 1 through 5. So cross that off. Do not worry about it until I can find where it's been relocated or a new one. We're not going to worry about doing this. Okay. All right. Moving on to the next part. We're going to go over collecting bloodstain patterns. Okay. So when it comes to collecting blood, all right. First off, if it's dry blood stain, it's all about getting your Q-tip, moistening it with some distilled water, and then rubbing it over, wait for that Q-tip to dry, and then you can package it in a clean paper bag. Once again, it's got to be breathable, so you can allow that DNA to stay as dry as possible, less chance of decomp. Now, this one also talks about how you can do tape-lifted bloodstains or scrape bloodstains into an envelope or something to that effect. If you're scraping blood off of a glass mirror or something, if you want to take it that way. Now, the last part is related to large bloodstains. If that is on clothing, bed sheets, carpet, if needs be, they'll cut it out. If not, they'll just take it and they'll package it up, and they will take it to an evidence locker where it actually will dry out. And then once again, they'll store it in any type of breathable, porous box, paper bag, paper wrapping, whatever it is. And then, of course, on any of those, you're going to seal it, you're going to label it, and put tamper-resistant tape, initial date. Always, always. All right. And this just, once again, talks about, well, when we're going to get into the different parts of bodily fluids detected. We're not going to worry about that. Okay. The next thing kind of finishes up the very end of your notes packet is the different careers related to serology and blood spatter. So the first one is a phlebotomist, and phlebotomists, their job is to collect blood samples. $12 an hour up to more, depends on where you work. It's in high demand. And this is just a license, like a certificate that you could receive. So it's a great way to pay your college by having this. It's kind of like being a CNA. So a phlebotomist withdraws your blood. The forensic serology DNA criminalist is the ones that test your bodily fluids, test whatever, if it's blood, if it's semen, if it's saliva. They do your genetic markers. They determine any type of blood typing, family connections that way. Now this salary is based off of Utah, and it can get up to 90 grand, but the base starts out at 32. Serology technician, like when you look at these, DNA criminalist, this is testing your blood, bodily fluids. And then you look at the serology technician, same thing when it comes to testing and analyzing different crime scenes. But these ones have a higher level of education. It's kind of like being the doctor versus the nurse. So when you're a serology technician, this is more of the doctor, and that's why you get paid more. All right. Now, kind of skip through this. The very last thing that I have on here is a little review. I'll pull this up for you. Luminol reacts with what? So this is just back in your notes, but luminol reacts with iron in the hemoglobin. Remember, the hemoglobins are on your red blood cells, so that's what luminol reacts with. Hydrogen peroxide releases what? It's an oxidation reaction, so it releases oxygen. And then what is the correct order of chemicals needed in order to perform the Kastle-Meyer test? And that's your alcohol, your phenolphthalein, and then your hydrogen peroxide that creates the oxygen for the phenolphthalein to turn pink. So that kind of sums it all up. Now, on the very front of your notes packet, you also have your little crossword. So just make sure you get that finished, and then you will turn this in Friday when you take the test. All right, you guys. Once you've finished watching this video, you can go through and look through the PowerPoint, anything you'd like to do that way, and then answer these 5 questions on campus. All right. I look forward to seeing you guys next week.